Hi everyone, this video covers some of the content from Key Area 4 from the Higher Human Biology course Unit 3. It just covers parts C, D and E, which is all about neurotransmitters and mood, and then how different drugs, medicinal and recreational, basically interact with neurotransmitters and affect the neural pathways. So here's the success criteria for the whole Key Area 4. Last week we had looked at 1 to 3, which were parts A and B of the Key Area. This week we're going to be looking at 4, 5, 6, 7 and 8. Now here are the SQA notes that basically outlines what the SQA want you to know about these different areas. So if you want, pause this video, have a little read through, maybe take some notes. I'm going to be expanding on these notes to basically go into greater detail, so hopefully you can understand it a little bit better. So as you can see, there's 3 slides there. There's quite a lot going on. We're first going to look at how neurotransmitters can affect mood and behaviour. Now, last video and last week we looked at the structure of neurons, or also known as nerve cells, and how 2 neurons can communicate with each other over the synapse using small chemicals called neurotransmitters. So we're going to be looking at endorphins and dopamine, so 2 types of neurotransmitters, often referred to as happy hormones or happy chemicals. In the diagram there'll also be other ones you might recognise, like serotonin and oxytocin, but we're going to be looking at dopamine and endorphins. So endorphins first of all. Actually, they're not neurotransmitters. They are chemicals that are like neurotransmitters, and they are basically involved in reducing pain. So without the endorphins, you'd be feeling a great bit of pain. So maybe you might be up on a scale at 9 or 10. And then when the endorphins are released, it's going to bring the pain down maybe to more moderate pain. And basically how they work is they bind to receptors blocking the pain signals, and they also stimulate a feeling of euphoria. So that is like intense sort of pleasure and happiness. Fun fact, SQA will say endorphins are neurotransmitters, but they're actually neurotransmitter-like chemicals. But for the principles of this, we're going to say they are neurotransmitters. Now, the endorphins are released in response to severe injury, which makes sense if we're trying to minimize pain and stop the pain pathway. They're also released through prolonged and continuous exercise. Now, this makes sense if you're feeling a bit low, or if you suffer depression or have any mental health problems. Often you'll hear, oh, go out for a walk or go do some exercise, because it does release those happy chemicals. Endorphins are also released during stress. They can be released by certain foods. So that's often why we like eating foods like chocolate and cake, because endorphins are released, so those happy chemicals. And they're also released through activities like sex as well. The other transmitter we're going to look at is dopamine, and it is involved in the reward pathway. So endorphins are involved in stopping the pain pathway, so minimizing pain, as well as increasing pleasure, and dopamine is involved in the reward pathway. It also induces feelings of pleasure, so happiness, and kind of makes you feel good. Okay, and it's basically released during certain activities, because it's your body's way of saying keep doing these activities. So if the activity is a benefit to you, dopamine will be released, make you feel good, so then you'll want to do it again. For example, if you eat when you're really hungry, it satisfies you, it feels really good, dopamine is being released. Okay, I'm sure, well, actually I don't know about you guys, but I can totally relate to this kid here who is clearly loving her watermelon. I'm very much linked to food and high dopamine releases. I do like food. So the way it works is when your need, if it is hunger or thirst, is satisfied, there's pleasure centers in the hypothalamus that release the dopamine. Now, if we look at this diagram here, we've got the hypothalamus just down there, and we've got some pleasure centers near there and in there that will release that dopamine. So these messages are taken to the frontal cortex, which is just here, so that you can actively appreciate that experience. So you're feeling those feel-good feelings. You're actively feeling good. So I've said feel good quite a lot there. So this is basically the reward pathway, and what the reward pathway actually is just the neural pathway that either secretes dopamine or responds to dopamine. And as I said, this pathway is activated when an individual engages in a behaviour that's beneficial to them. And the whole point of this is it's an evolutionary response. We've evolved to release the dopamine so that it encourages us to repeat beneficial behaviour. Okay. Now here is a more detailed diagram of the reward pathway. So we start in here with our hypothalamus, and the dopamine messages are going to be sent up to here, and this is our frontal cortex. So it's hitting areas that have got things to do with judgment, thinking, and willpower. So that, you know, that way where you just can't stop yourself going for another chocolate or finishing the giant bag of crisps because that dopamine hits so high. You think, oh, it feels so good. So it's affecting this bit about willpower. We're also touching on cravings there as well. And then we're also going to be stimulating the area that actually allows us to focus on what is the source of temptation, what is actually giving us that feel-good hit. So now we're going to look at neurotransmitter-related disorders, and these are basically diseases or disorders that affect either the production of neurotransmitters or the response to neurotransmitters. Now if you look at this diagram, this one here that I've circled is the normal one. So it's showing a presynaptic nerve releasing a normal amount of neurotransmitters and a postsynaptic nerve with a normal amount of receptors. Now if we look at our B diagram here, what we've got is a reduced number of neurotransmitters. So this individual who's got these neurons will be deficient in a neurotransmitter, because as you can see, the receptors are not the issue, they've got enough receptors. However, in this individual, we've got plenty neurotransmitters, so the individual's actually deficient in receptors. So we've got a reduced receptor here. So in B and C, these individuals have a neurotransmitter related disorder. Now I'm just going to talk about a few of these disorders, just so you can put it into context, you know what we're talking about. Things like Alzheimer's, epilepsy, Parkinson's, anxiety, depression. These are all conditions that are associated with deficits in neurotransmitters or their receptors. I'm just going to go over a few of these conditions in case you've heard of them, but you're not really too sure what they are. So Alzheimer's is a degenerative disease, so it gets worse with age, and it affects brain cells and causes problems in memory, thinking, and behaviour. And this is because of a loss of the neurotransmitter of acetylcholine. Okay. Now, epilepsy is basically a disturbance in the normal pattern of neuron activity and can result in seizures and fits and epileptic fits and things. Now what happens here is it's actually an inhibitory signal that is disrupted. So GABA, the neurotransmitter GABA, is actually used to turn off impulses. Now, people that are epileptic have a reduction in GABA. Oh, sorry, I'm going to circle that. A reduction in GABA. This means that neural pathways aren't getting turned off when they're meant to be turned off, resulting in that really sporadic fitting. Okay. Next one is Parkinson's, another progressive nervous system disorder, and this affects the movement. And what we've got here is it's basically loss of neurons that are actually producing dopamine. Now, there are lots of drugs that can treat neurotransmitter-related disorders, and these drugs will interact with the neurotransmitter pathways that are affected. So we're going to be looking at three sort of categories here: agonists, antagonists, and inhibitors. And within the inhibitors, we're going to look at enzyme inhibitors and reuptake inhibitors. Now, I'm going to go over each of these. A lot of people can get them confused. So if you're thinking, oh, I've seen this before, because you're part of Belardo's Higher Human Biology team, it's because I went over this in the live lesson. It's just because a lot of people find it confusing, so we're just going into detail with it twice. Okay? So, agonists and antagonists. These two types of drugs interact with the receptors on the postsynaptic membrane. So, these are the receptors that the natural neurotransmitters will come and bind to and normally initiate a response if that's appropriate, or in GABA's situation, not initiate the response, to stop the responses. So agonists and antagonists interact with the receptors. An agonist, agonistic, these are basically ones that work with the natural neurotransmitters, they mimic them. And antagonistic is the opposite, so it's stop. So if you think about it, if somebody is behaving antagonistically towards someone, they're sort of working against them, they're causing conflict. So an antagonistic drug is one that works in conflict with the natural neurotransmitter, whereas an agonistic drug is one that works with it. So it's go, it's a go situation. It's wanting the natural neurotransmitter to be enhanced and keep working. We're going to look at agonists first. Now, agonists are basically chemicals that bind to the receptor on the postsynaptic membrane and enhance the response from the neurotransmitter. So if the normal neurotransmitter bound to the receptor, something would happen. What an agonist does is it enhances this response. It basically increases it. It does this by mimicking the naturally occurring neurotransmitter. And if we look at this graph here, what we've got up this side is activity of postsynaptic neuron, and along the bottom is our concentration of the drug, or whether the drug's there or not. So if we have no drug, it's this black line, it's just constant. Now, if we give them something that works as a full agonist, then we can see that as soon as we give the full agonist, the activity increases. Okay? And as the concentration of the full agonist drug increases, our activity increases up to a point, well, up to 100%, and then it can't get any better. Now, a partial agonist is another type of agonist, but it's kind of like half full or half the power of agonists. So it's not completely mimicking the natural neurotransmitter, it's just partially mimics it. So this is why it just increases half the way and not the full way. But the point of this is that agonists are chemicals that mimic the natural occurring neurotransmitter to enhance their action. And you can see that if you get given an agonist, the activity in the postsynaptic neuron will increase. Just as a side note, agonists are used to treat things like Parkinson's. Now, here is a good diagram to actually show the action of an agonist. So on this side, we've got the normal situation. We've got our receptor sites down here, and we've got our naturally occurring neurotransmitter. And as you can see, the neurotransmitter fits very nicely into the receptor, and then an impulse is generated, if that's the appropriate response. Remember, some are inhibitory, so no impulse will be responding, which is why we've got normal cellular activity rather than impulse, because it might be that impulse is not the normal cellular activity. So anyway, point is, normal situation, neurotransmitter fits in receptor, triggers whatever reaction is appropriate. Now when you've got an agonist here, notice that the bottom of it is a similar shape to our neurotransmitter. This means that the agonist can fit very nicely into the receptor, and because it's mimicking it and it's similar to the neurotransmitter, we're actually going to enhance the cellular activity. So we're going to be increasing the normal response that the postsynaptic neuron would give. So agonists, similar shape, similar chemical makeup to basically mimic the neurotransmitter and enhance that response. So now we'll look at antagonists. So antagonists are the ones that work against the neurotransmitter. These work by binding onto the receptors on the postsynaptic neuron, and they actually block the action of the normal neurotransmitter. This will either reduce the transmission of nerve impulses or stop them altogether. So I've got the same graph here as before, but now we're focusing down here. I've added a new line, which is our antagonist. Now, what we can see is as the concentration of antagonist increases this way, our activity of the postsynaptic neuron starts to decrease. And if we've got high enough concentration of antagonist, the activity actually stops altogether. Okay? And the way this works is by blocking receptors. I'll show you on the next slide. But just as a side note, antagonists are used to treat anxiety disorders. So if you imagine anxiety is, people are overthinking, things are happening really fast in your head. If you've ever been anxious, it's kind of like things going, and basically what an antagonist does is stops those neural pathways. So you can slow down and there's not as many impulses getting fired or slowing everything down. So that's what an antagonist would do to treat an anxiety disorder. Right, here we've got same diagram before, same on the normal side, neurotransmitter fitting into receptor site, but our antagonists are a different shape. They fit into the receptor and they block it, but they do not actually trigger that receptor. So what we've done is we've just blocked it completely, so all cellular activity is blocked. So any impulses that would have been triggered are obviously now not triggered. The last one that we're going to look at are inhibitors. Okay, so we've got agonists and antagonists. They interact with the receptors on the postsynaptic neuron, but inhibitors, they don't interact with the receptors. Okay, they interact with other molecules that influence the presence or absence of the natural neurotransmitter. Okay, now two types we're going to look at: enzyme inhibitors and reuptake inhibitors. Before we go into them, we need to have a little reminder. We talked about enzymes and reuptake channels at the start of Key Area 4, so in last week's video. Can we remember what they actually do? What do the enzymes do? What role do they play in neurotransmission, and what role do the reuptake channels play? Think of their connection to neurotransmitters. What are they actually doing? Pause this video, have a think. Right, you're back. So here's our little recap if you haven't worked it out. Neurotransmitters are removed from the synaptic cleft by degradation by enzymes, so the enzymes will degrade the neurotransmitters, or the neurotransmitters will go back into the presynaptic neuron through a reuptake channel. Now, if we look at this diagram, we've got our presynaptic neuron here and our postsynaptic neuron there. Now, the neurotransmitters get released into the synaptic cleft here, and basically they diffuse over, bind to the receptors, and then initiate the appropriate response. Now, in normal situations, if those neurotransmitters are not removed, we're going to be continuously stimulating that postsynaptic neuron. So we're going to be constantly firing off impulses. In normal situations, that's not good because you want to be able to control whether you're firing them or not. So once you've sent your message, you want to stop stimulating the postsynaptic neuron. So to do that, we need to remove the neurotransmitters. And that's what these enzymes do here. They will break down the neurotransmitter to stop the stimulation of the receptor, or the neurotransmitter will go back through the channel and so it can get reused later. Now, if you've got a disorder where you are deficient in a neurotransmitter, so maybe you're not producing enough of a neurotransmitter, the way we can target that is to stop the removal of the neurotransmitter. So we'll look at enzyme inhibitors first. If we can inhibit the enzyme that degrades the neurotransmitter at the synaptic cleft, then this means that the concentration of the naturally produced neurotransmitter isn't going to decrease. So that means that neural pathway will continue to be activated. So this will help target stuff where you're not producing enough of a neurotransmitter, or you need the pathway to be a bit more continuous, to be turned on a bit more. Enzyme inhibitors are used to treat things like Parkinson's. Because if you remember, Parkinson's, you're deficient in dopamine. So if you're deficient in dopamine, if we remove the enzymes that break dopamine down, then the dopamine concentration will increase to a more normal level. The other inhibitor that we're going to look at is a drug that inhibits the reuptake of the neurotransmitter. So as we said, the neurotransmitters can get taken back into the presynaptic neuron through a channel, which is normally here, but a reuptake inhibitor will actually come and block that channel. That means that the neurotransmitters can't go back into the presynaptic neuron, which of course means that they're going to remain in the synaptic cleft, so the natural effect of the neurotransmitter is enhanced. Same thing as before, we're increasing the concentration of the natural neurotransmitter by not removing them. And one of the conditions that it can be treated with reuptake inhibitors is depression. Okay, so if you imagine depression, you've not got enough of those happy chemicals. What do? Well, we stop the happy chemicals like serotonin or dopamine being removed from the synaptic cleft, so those neural pathways, the happy neural pathways, keep getting triggered. Okay. The other thing we're going to look at is recreational drugs. So the first drugs that we were talking about, we're talking about treating conditions that a doctor might diagnose and then might advise you on what drugs to take. Some people take other drugs recreationally. And I'm not just talking about illegal stuff. Recreational drugs can also refer to legal things, so like caffeinated drinks, energy drinks, like cigarettes, alcohol. All of these have got chemicals in them that will affect your mood and behaviour. And of course, we're also going to be talking about things like illegal drugs like marijuana, ecstasy, heroin, massive list. Okay. Now these recreational drugs, many of them act as agonists or antagonists. So they're basically interacting with the natural neural pathways in your brain to stimulate feelings of pleasure. So recreational drugs affect the neurotransmission in the brain at the synapses in a number of ways. They can alter mood, so they can make you really happy, but at some point, some drugs will actually then bring you back down again. So it's not always a positive effect. Sometimes a negative effect will follow a positive effect. They can affect your cognition, so that's how you think, decisions you might make. Perception, so how you perceive things, like how you perceive things that are going on. And also behavior as well. So they can maybe make you more spontaneous, or lower your inhibitions, so that you can partake in more dangerous activity. Many recreational drugs affect the reward pathway of the brain. Okay, so remember we're talking about dopamine, that neurotransmitter that's one of the happy chemicals. And what we've got here in this this sort of diagram here is we've got our dopamine. Where's my pen? We've got our dopamine, that's these little star-like shapes. And in the second side, we've got these red bits, which is cocaine. So that's a recreational drug. And the way cocaine works and why it makes people feel good is that cocaine is actually one that's going to come in and block the reuptake of dopamine. So if you're blocking the reuptake of dopamine, that means the dopamine will be around the synaptic cleft to keep binding onto those receptors and sending those sort of pleasure pathways, making you feel good. Although I've just said a lot of things that make drugs sound good, they make you feel good and stuff, there's actually a lot of really bad side effects that can be very dangerous, which is why a lot of these drugs are illegal, because they can cause deaths if used incorrectly, or if you take a dodgy chemical or something, maybe one that's not quite right. Now, repeated use of drugs can lead to two things: drug addiction and drug tolerance, or drug tolerance, okay? And they're two quite different things. So drug addiction is basically when an individual craves more of the drug. Their body really needs it and wants it. And this is a result from something called sensitization. Now, drug tolerance is a bit different. It's a result of desensitization. And this is basically when an individual has to take more of a drug to obtain the same effect. So, for example, if an individual used to have to take one pill to feel really good, over time they're going to have to start to take 2 pills to feel just as good as they used to. And then over a longer time they'll have to take 4 pills just to feel the same high that they were used to feeling before. Their body's becoming tolerant of the drug. Now we're going to look at drug addiction in greater detail now. Drug addiction is associated with antagonistic drugs. So these are the ones that work against the neurotransmitters, the naturally occurring neurotransmitters. So the antagonists are ones that are going to block the receptors and stop the transmission of a natural neurotransmitter. Now they can lead to drug addiction through sensitization. So one of the effects of repeatedly using an antagonist is the nervous system adapts and changes. And what it does is it basically increases the number of receptors of the natural neurotransmitter that the antagonist is normally blocking, and it also increases the sensitivity of those receptors. So, if there are more receptors that are more sensitive to the drug, then that means that with repeated drug taking, we're going to increase the effect of the drug, okay? Because the more drug you take, the more sensitive the receptors become and the more receptors there are. This means that the actual effect of the drug is going to increase over time. So if we have a look at this graph here, as our dose increases, okay, the effect of the drug, as normally before you were addicted, increases to a certain degree as well. As we can see, that's our black line there. But after sensitization, because we now have more receptors that are more sensitive to the drug, we actually increase the relative effect, okay? So if you took, I don't know, say 3 pills before you were addicted, you'd get this effect. But if you take 3 pills after you're addicted, because you've got more receptors and they're more sensitive, your effect increases. So the effect of the drug will actually increase. And essentially, this means the feel-good effect is getting amplified. So the person starts to crave more of the drug because it feels so good. They're wanting it more and more and more. And that's where their addiction is coming from. Now, drug tolerance is associated with agonists. So drug addiction is associated with antagonists, and drug tolerance is associated with agonist drugs. Okay, if you remember, an agonist is one that mimics the natural neurotransmitter, so they're going to enhance the effect. Okay, so by repeatedly using an agonist, it's making your body think that it's producing lots of the naturally occurring neurotransmitters. So your body is going to be turning on those neural pathways more than it should. So in response, your body then decreases the number of receptors and decreases the sensitivity of these receptors. And this leads to desensitization, and that is basically drug tolerance. Because if you've got less receptors to a neurotransmitter, and these receptors are less sensitive to the drug, then that means that the individual is going to have to take more and more of the drug to actually get the effect. Okay, so if we look at this graph, we've got relative effect up the side, and we've got increasing dose here. Now, before you're tolerant of the drug, you're looking at the black line. So as concentration increases, effect increases. When you become tolerant of a drug, you still get the same relationship as concentration increases, effect increases, but the relative effect will decrease. So again, imagine you had to take 3 pills to get a certain buzz before, and maybe we're here. We take 3 pills to get this level of a buzz. When you get drug tolerance, when your tolerance increases, you might have to take 5 pills to get to the same level. Okay, so before you're drug tolerant, you need to take this concentration, but now you've got drug tolerance, you actually need to take a higher concentration, but to get the same effect. Okay, so what this really results in is a person is taking more and more each time they take a drug to get the same good effect or feel good effect that they had before. So now we're going to combine these graphs. We've got our drug addiction v drug tolerance. So drug addiction, let's do it in red, is the sensitization. And then we've got the drug tolerance in purple here. So drug addiction will increase the effect of a drug, making your body want to crave it. And drug tolerance will decrease the relative effect, meaning that you've got to take more of that drug to get the same effect as before. Now, many of these recreational drugs don't just work one way. They're actually going to work a number of different ways. So they'll be antagonistic to some neurotransmitters and agonist to other neurotransmitters. So you can get situations where you are addicted as well as tolerant. So it's not always a simple clear cut between the two. So hopefully that has helped you to understand how neurotransmitters can affect behaviour and also how different drugs will interact with the pathways that neurotransmitters work along. If you've got any questions, just ask in the comments below. But of course, if you're from Balerno, from the Higher Human Biology pupils, just tag me on Teams and I'll get back to you. Now, if you liked the video, please give it a wee like because it makes me feel good. If you didn't like the video, keep that to yourself. Thanks. Right, I'll see you later. Bye.